It's kind of mind-boggling when you realize just how much of your lifelong health story is written in those first 1,000 days. From the moment you're conceived until you're about 2 years old. Talk about laying the groundwork.
It truly is a foundational period. Think of it as this incredibly sensitive window where your environment is actively sort of communicating with your biology, setting patterns that can influence your health for, well, decades to come.
And that's precisely what we're plunging into today. We're taking a deep dive into how this very early environment, this early terrain, as we're calling it, shapes a person's health. We'll be exploring the fascinating interplay of your microbiome, that vast community of microbes living in and on us.
Yeah, that whole ecosystem.
Exactly. And epigenetics, which is all about how your genes are expressed, right? Not the genes themselves, but the expression.
Precisely. And for this exploration, we've drawn from a wide range of research. We've looked at studies on early human development, the intricate world of our microbial inhabitants, and the dynamic field of epigenetics and how our environment interacts with our genetic blueprint.
Our mission for you, the learner, is to really get a handle on these critical factors that mold your long-term well-being right from the start. We want to give you those essential aha moments without you feeling like you're, drowning in technical details.
And to keep our journey clear, we'll be navigating through three key areas. First, how we inherit and establish our initial microbial communities. Second, the remarkable flexibility and programming power of our epigenetics. And third, how all of this lays the foundation for our resilience in the years ahead.
Okay, let's peel back the layers on that first key area. Microbial inheritance and colonization. It all kicks off right at birth with what some researchers have kind of poetically termed the bacterial baptism.
That's a vivid way to put it, and it's incredibly apt. Your very first encounter with the microbial world is, well, profoundly significant. The types of microbes you're exposed to initially differ dramatically depending on how you enter the world.
Right. If you're born vaginally, it's like being bathed in your mother's vaginal and fecal microbiota, a rich mix of bacteria like lactobacillus, prevotella, and bacteroids. It's like nature's tailored initiation.
So the crucial insight here is that your very first microbial exposure sets a distinct trajectory for your gut health.
And in contrast, babies born via cesarean section, they get a different set.
That's right. They're primarily introduced to the microbes present on the mother's skin bacteria, such as Staphylococcus, Corynebacterium, and Proponobacterium, as well as, whatever microbes are circulating in the hospital environment.
And this different starting point has real consequences, doesn't it? I remember reading studies showing C-section babies often have lower initial diversity in their gut microbiome.
That's correct, and often a delay in the colonization of those beneficial bacteroids, too.
Interesting. And what's truly fascinating is the work of Dr. Maria Gloria Dominguez-Bello. She's been really central in characterizing these initial microbial differences and even pioneered the concept of vaginal seeding.
Ah, yes, vaginal seeding. For anyone unfamiliar, that involves transferring some of the mother's vaginal fluids to a baby born by C-section shortly after birth.
The aim being to mimic that natural microbial exposure they missed.
Exactly. And, well, initial smaller studies did suggest some promising results, indicating a potential partial restoration of the oral, skin, and even gut microbes in those infants.
But it's so important to emphasize that this is still a very active area of research. The big question mark is safety, right?
Absolutely. We need to be absolutely sure we're not inadvertently transferring any harmful pathogens. That's why rigorous screening protocols and large-scale clinical trials are essential.
And haven't more recent, larger studies actually shown less of an impact than initially hoped?
Yes, indeed. Some larger, more recent work hasn't found significant long-term impacts of vaginal seeding on the infant's gut microbiota composition, or even their risk of developing allergies later on. It really highlights just how intricate and complex the microbial world is, and frankly, how much we still have to unravel.
Okay, so moving beyond the moment of birth itself, it seems that early physical contact is also a really important factor. I'm thinking specifically about skin-to-skin contact immediately after birth.
Absolutely crucial. Skin-to-skin contact isn't just about that beautiful bonding experience. It actively facilitates the transfer of the mother's skin microbiota to the infant. And while it might not be the primary driver of the gut microbiome's initial composition,
It has other knock-on effects.
precisely, it has a cascade of other benefits supporting successful breastfeeding, reducing stress levels in the baby, stabilizing their physiological systems, all of which indirectly contribute to the establishment of a healthy microbial ecosystem.
And then there's the whole universe of microbes within the household itself. Family members, our beloved pets, even the dust lurking in the corners.
Exactly. The infant's microbial world rapidly expands to encompass all of that. Siblings, for example, can be a surprisingly significant source of microbial exchange.
It's incredible to think of our homes as their own miniature ecosystems. Wasn't there a project on that?
Yes. Dr. Rob Dunn's Wildlife of Our Homes project really brought that to light. The sheer variety of microscopic life within our dwellings is quite astonishing.
Wow.
And Dr. Jack Gilbert's Home Microbiome project took this understanding even further, demonstrating that we humans are the primary architects of our home's unique microbial signature. When a family moves into a new place.
Their microbial fingerprint follows them pretty quickly.
Remarkably quickly, yes. And our furry companions play a role in this too, don't they? Especially those dogs and cats that venture indoors and outdoors.
They certainly do. They tend to enrich the microbial diversity within the home by introducing bacteria associated with soil and plants, things they bring in from outside.
Even something we often overlook, like household dust, turns out to be a reservoir and a kind of living record of the entire indoor microbial environment.
So it's like a layered process. You get the initial dose from mom at birth.
Right, the initial maternal transfer, followed by this continuous shaping and modulation influenced by the family members and the environment of the home itself.
Okay, so now, what happens when this initial microbial seeding is delayed or altered, as we often see with C-section births? Are there long-lasting health consequences?
Unfortunately, the evidence suggests that yes, there can be. The mode of birth and those very early microbial exposures have been linked to health implications that could extend throughout life. We observe a correlation between altered initial seeding, particularly in C-section births, and an increased risk of developing non-communicable diseases later on.
What kind of diseases are we talking about specifically?
Conditions such as asthma, various allergic conditions, both food and respiratory allergies, atopic dermatitis, eczema, celiac disease, type 1 diabetes, and even obesity and inflammatory bowel disease.
Wow, that's quite a list. It's as if these early microbial experiences create what some researchers refer to as lasting immune gaps.
That's a very insightful way to describe it. Early microbes are absolutely fundamental for the education, calibration, and overall maturation of the immune system. They play a vital role in training immune cells to distinguish between friend and foe, in establishing tolerance to harmless substances we encounter.
Like food or pollen.
Exactly. And in carefully balancing the body's pro- and anti-inflammatory responses.
So if those initial microbial signals are missing, delayed, or qualitatively different, the immune system doesn't receive that crucial early instruction. It's like missing the first day of school.
Precisely. This can lead to a predisposition for the immune system to overreact to things that are normally harmless, or to fail to effectively recognize and combat genuine threats. Even the specific types of bacteria we encounter in those very early moments seem to differentially train the nascent immune system.
It really underscores just how critical the timing and the nature of those very first microbial encounters are. That critical window in early life truly has a profound and lasting impact.
Which is why there's such significant public health interest in strategies to optimize early microbial seeding. This includes supporting vaginal birth when it's medically safe, using cesarean sections judiciously, promoting immediate skin-to-skin contact, and early initiation of breastfeeding.
And maybe even thinking about our home environments.
Right, considering how our home environments can foster beneficial microbial diversity through things like having pets, connecting with nature, and practicing, mindful hygiene, not over-sterilizing everything.
Okay, so after this initial microbial baptism and the profound influence of the early environment, the next really critical piece of the puzzle seems to be how we nourish this brand new ecosystem. And breastfeeding appears to play a starring role here, doesn't it? It's clearly so much more than just providing calories.
Absolutely. Breastfeeding is a dynamic and multifaceted process that actively shapes the infant's developing microbiome and their intricate immune system. It's really a continuation of that early terrain programming we've been discussing.
And one of the real unsung heroes in breast milk seems to be human milk oligosaccharides, or HMOs. I find these fascinating.
HMOs are truly remarkable molecules. They are complex carbohydrates and are actually the third most abundant solid component found in human milk after lactose and lipids. What's even more remarkable is the sheer diversity of their structures. Over 200 different ones have been identified so far.
200. Wow, that's an incredible variety. And the interesting thing is babies can't even digest them directly, right? They're not for the baby's energy.
That's correct. They are indigestible by the infant's own digestive enzymes, but they serve as a highly selective food source, a potent prebiotic, you could say, specifically for beneficial bacteria residing in the infant's gut.
Like those incredibly important bifidobacterium species, the ones we want to encourage.
Exactly. Specific strains like Bifidobacterium infantis and Bifidobacterium brave possess specialized enzymatic machinery that allows them to break down and thrive on these HMOs. So in essence, HMOs selectively nourish these bifidobacteria, helping them to become the dominant players in the gut microbiota of breastfed infants.
So the key takeaway here is that HMOs are specifically feeding the good guys, acting like a personalized fertilizer for the baby's gut garden.
That's a great analogy. And HMOs likely do more than just act as prebiotics, don't they?
Right. They have other jobs too.
They do indeed. They have direct protective effects as well. They can act as decoy receptors, binding to pathogens and preventing them from adhering to the delicate lining of the baby's gut. They also play a crucial role in the maturation of the gut barrier itself and directly influence the development of the infant's immune responses.
And with all those diverse HMO structures you mentioned, it sounds like they could potentially have a really wide range of different effects depending on the specific type.
Precisely. Emerging research is continually revealing that different HMO structures may have distinct impacts on various aspects of infant health, ranging from gut health and immune system development to potentially even influencing brain development and cognitive function. It's a remarkably sophisticated and tailored prebiotic effect.
And then there's the transfer of maternal IgA antibodies through breast milk. That sounds like another critical way that breastfeeding provides protection.
Yes, secretory IgA, or SIGA, is the predominant type of antibody found in breast milk, and it provides invaluable passive immunity, specifically at the mucosal surfaces of the infant, particularly in the gut.
And this isn't just a generic antibody, is it? It's almost like it's customized to the baby's environment.
Exactly. There's this fascinating connection, the enteromammary and bronchomammary links, where B cells, a type of immune cell, in the mother that have been exposed to pathogens in her own gut and respiratory tract actually migrate to the mammary glands. And there, they produce IgA antibodies that are specifically targeted to those same threats that the mother and baby are likely to encounter in their shared environment.
So the baby receives antibodies that are literally designed to fight off the specific bugs they're most likely to encounter right then and there.
Precisely. And this IgA is actively transported into breast milk in a stabilized form known as SIGA, which is resistant to digestion in the infant's gut. Its primary mode of action is immune exclusion. It binds to those pathogens, preventing them from attaching to and invading the infant's tissues, all without triggering excessive inflammation. It's a very targeted and gentle form of protection for the newborn.
And breast milk is just brimming with other bioactive components as well, right? It's like a whole pharmacy tailored for the baby beyond just the HMOs and IgA.
It truly is. You have lactoferrin, which has both antimicrobial properties and helps to modulate the immune system. A diverse array of cytokines, which are signaling molecules that can be both pro and anti-inflammatory and help to train the infant's immune responses. Various growth factors that contribute to the maturation of the gut itself.
Like the gut lining.
Yes, the epithelium and the lymphoid tissues associated with the gut, as well as influencing the development of immune cells. And then you have other components like lysozyme, milk lipids, hormones, all with their own antibacterial, anti-inflammatory, and developmental effects. There are even maternal cells like leukocytes present in breast milk that may contribute to the baby's developing immunity and tolerance.
It sounds like all of these components work together in this incredible synergy, like a constant dialogue happening between the diet, the microbes, and the baby's own developing immune system.
Exactly. And because the maternal SIGA antibodies reflect the mother's unique exposure to pathogens and the HMO profiles can vary between mothers, it truly is an incredibly personalized form of medicine and essential developmental support.
Which really underscores why promoting breastfeeding is such a critical public health intervention.
Absolutely. And why replicating its intricate complexity in infant formula remains such a formidable challenge.
Okay, so we've explored the initial microbial seeding and the vital role of breast milk in nourishing that early ecosystem. Now, let's turn our attention to our third key area, epigenetics, and how those early life experiences leave a lasting imprint on how our genes are expressed.
Epigenetics. It's a fascinating and rapidly evolving field, It's essentially the study of heritable changes in gene expression that don't involve alterations to the underlying DNA sequence itself.
So not changing the genes, but changing how they're read.
Exactly. Think of it like switches or dimmer controls that can turn genes on or off or turn their activity up or down. The primary mechanisms we're learning about involve things like DNA methylation, adding little chemical tags to the DNA histone modifications, changing how the DNA is packaged, and non-coding RNAs. Essentially, early life experiences can flick these switches, changing gene activity for the long term, even without altering the DNA code itself.
And it sounds like the first 1,000 days are a period of really remarkable epigenetic plasticity, meaning things are very adaptable and responsive to the surrounding environment during that time.
Precisely. During this incredibly sensitive window, the epigenome is highly susceptible to environmental cues, and these epigenetic modifications can essentially translate those early life experiences into long-lasting biological memories that can significantly shape our health trajectory throughout life.
And it seems that maternal influences during pregnancy and in those early months of infancy play a really significant role in sculpting the infant's epigenome. Let's start by looking at maternal diet and nutrition. How important is that?
Absolutely crucial. Optimal maternal nutrition provides the essential building blocks, the necessary cofactors and substrates for the enzymes that drive DNA methylation and histone modifications. Key nutrients like folate, vitamin B12, choline, methionine, and zinc are all vital players in these processes.
So deficiencies or even excesses could throw things off.
Exactly. Deficiencies or excesses of these nutrients during those critical developmental windows can significantly alter the baby's epigenome and potentially increase their risk for developing metabolic disorders, cardiovascular disease, and other chronic health conditions later in life.
And it's not just about the nutrients themselves, is it? Maternal stress, trauma, and the mother's overall mental health during pregnancy can also leave their mark on the developing baby's epigenome.
Yes, significant maternal psychological stress, anxiety, depression, or experiences of trauma during gestation can lead to epigenetic modifications in the offspring, often in genes that are involved in regulating the body's stress response system, like the hypothalamic pituitary adrenal, or HPA axis.
And Dr. Rachel Yehuda's research has provided some really compelling human insights into this, hasn't it? Looking at the children of Holocaust survivors and mothers who were pregnant during 9/11.
Her work has been groundbreaking. It's yielded crucial human evidence supporting this idea of intergenerational transmission of trauma-associated epigenetic marks. Her team particularly focused on genes like NR3C1, which codes for the glucocorticoid receptor, a key part of the stress response, and FKBP5, which regulates the sensitivity of that receptor.
And they found actual changes in methylation.
Yes, they found altered DNA methylation patterns in these specific genes linked to the stress exposure, and importantly, observed similar patterns across generations.
Wow. It's also becoming increasingly clear that the mother's own microbiome, her gut bacteria, can have an influence on the infant's epigenome too. How does that work?
It's a really interesting connection. The various metabolites produced by the mother's gut microbes, such as short-chain fatty acids like butyrate, which can act as an HDAC inhibitor, affecting histone modifications, as well as essential nutrients like folate, which is a methyl donor.
Those can get to the baby.
Exactly. They can cross the placenta during pregnancy or be transferred through breast milk after birth. These molecules can directly interact with and influence epigenetic processes in the developing infant. So if the mother has an imbalanced gut microbiome or dysbiosis, it could indirectly lead to alterations in the epigenetic landscape of the baby.
And of course, exposure to environmental toxins during pregnancy is another factor we need to consider.
Yes, absolutely. Exposure to various pollutants, endocrine-disrupting chemicals, cigarette smoke, specifically nicotine and alcohol during pregnancy, can all induce epigenetic modifications in the fetus with potentially detrimental long-term health consequences for the child.
So the epigenome really acts as this critical intermediary, translating all these early environmental exposures, diet, stress, microbes, toxins, into potentially lifelong alterations in gene expression and physiological function.
That's a great way to summarize it.
You mentioned NR3C1 and FKBP5 as key stress-regulating genes. Let's delve a little deeper into how these specific genes might be epigenetically modified. What happens with NR3C1, the glucocorticoid receptor gene?
Okay, so NR3C1 is particularly interesting because it plays such a pivotal role in the HPA axis, our central stress response system. The promoter region of this gene, which essentially tells the cell how much receptor to make, is a key site where DNA methylation can occur. Increased methylation in this region is often associated with reduced expression of the glucocorticoid receptor.
And why is that significant for our stress response? Fewer receptors means?
Fewer receptors means the negative feedback loop that helps shut down the cortisol response becomes less effective.
Mm.
This can lead to a prolonged release of cortisol after a stressful event, a heightened overall stress reactivity, and potentially an increased susceptibility to developing mood and stress-related disorders later in life.
And studies have linked mom's stress to these changes in the baby.
Yes, studies have indeed linked maternal prenatal stress, depression, or anxiety levels to altered NR3C1 methylation in cord blood, the placenta, and infant tissues. And these changes have, in turn, been associated with how those infants respond physiologically to stressful situations and their subsequent neurobehavioral development, things like temperament and cognitive abilities. The timing of the stress exposure during gestation also seems to be quite important.
Okay, and what about the FKBP5 gene? How does that fit into the picture?
FKBP5 is another key player. It provides the instructions for making a protein that actually regulates the sensitivity of the glucocorticoid receptor. Interestingly, exposure to stress can induce changes in the methylation patterns of the FKBP5 gene itself. For example, demethylation, the removal of methyl groups in certain regions of FKBP5, can lead to increased expression of the FKBP5 protein.
And more FKBP5 protein makes the receptor less sensitive.
Exactly. Increased FKBP5 can make the glucocorticoid receptor less sensitive to cortisol, which, again, can lead to a prolonged and potentially dysregulated HPA axis response to stress. Dr. Yehuda's research on Holocaust survivors and their offspring showed altered FKBP5 methylation patterns in both generations, and these patterns correlated with their baseline cortisol levels, suggesting that FKBP5 methylation might be involved in the intergenerational transmission of vulnerability to stress.
It really highlights how this specific type, the precise timing, and the overall duration of maternal stress or nutritional challenges can lead to these very specific epigenetic signatures in the child with potentially long-lasting functional consequences. It really underscores the importance of those critical and sensitive windows of development in the first 1,000 days.
Absolutely. And then there's the even more profound and, frankly, somewhat mysterious concept of transgenerational epigenetic effects.
Great, this is the idea that epigenetic changes can be passed down even further than just parent to child.
That's the idea. It suggests that epigenetic modifications induced by ancestral exposures, things like trauma, famine, or chronic stress in a parent, or even a grandparent, the F0 generation, can somehow be transmitted through the germline, the sperm or egg cells, and affect the health and characteristics of subsequent generations like the F2 or F3, even if those later generations haven't directly experienced the initial exposure themselves.
That's truly mind-bending. So experiences our great-grandparents had could potentially have a subtle biological echo in us today through epigenetics.
That's the current understanding of the premise, yes. It suggests that early life programming can have effects that ripple through family lineages, potentially contributing to patterns of disease risk or resilience that we observe across generations. The mechanisms are still being actively investigated, involving DNA methylation, histone states, non-coding RNAs in the germ cells. It's complex.
And the microbiome likely plays a role in this intergenerational epigenetic story as well, connecting back to what we discussed.
It certainly seems plausible. Remember those microbial metabolites we discussed, like butyrate and folate? They have the capacity to directly influence the epigenetic machinery within our cells. So, if the maternal microbiome is altered due to factors like diet or stress, it could indirectly program the infant's epigenome by changing the availability of these key metabolites, either during pregnancy, through the placenta, or after birth via breast milk. It's all incredibly interconnected.
It really underscores how vital it is to prioritize interventions that support maternal well-being during both pregnancy and the postpartum period. Things like ensuring good nutrition and providing support for stress reduction for healthy infant epigenetic programming.
Definitely. And a deeper understanding of these complex gene-environment microbiome interactions could pave the way for really targeted nutritional or microbial interventions in the future, potentially personalized approaches.
Now, let's turn our attention to the fascinating and increasingly recognized connection between the community of microbes in our gut and our brain, what scientists refer to as the microbiota gut-brain, or MGB axis.
Yes. And the first 1,000 days of life are absolutely pivotal for the establishment and fine-tuning of this crucial communication network.
This is where we're starting to see that the gut microbiome isn't just involved in digestion. It seems to have a real influence on things happening way beyond the gut, including our mental well-being. It's like our gut and brain are in constant conversation.
Precisely. It's a bidirectional communication network involving neural pathways, hormonal signals, immune system interactions, and metabolic exchanges. And those early microbial colonists play a fundamental role in setting up and calibrating this axis. The implications for lifelong mental and metabolic health are profound and long-lasting.
And we're seeing more and more compelling research that's linking the composition and diversity of the infant gut microbiome to later neurodevelopmental outcomes, right? Things like cognitive abilities and emotional regulation.
It's exactly. Several studies have identified associations between the specific types of bacteria present in an infant's gut at around 1 year of age and their cognitive performance at age 2. For instance, some research has linked a higher abundance of Bacteroides bacteria to better language development scores in toddlers.
But I also recall reading some research that suggested that the relationship between overall microbial diversity and neurodevelopment in early infancy might be a bit more complex than just more diversity is always better.
That's a very astute observation, and you're right. There have been some seemingly paradoxical findings where higher overall microbial diversity in the gut at 1 year of age was actually associated with slightly lower scores on certain cognitive scales at age 2.
Huh. Why would that be?
Well, it highlights that it's likely not just about having a large number of different microbes, but rather about having the right kinds of microbes in the right proportions and at the right times during those critical early developmental windows. It's possible that in very early infancy, a less complex, more specialized microbiome, like the bifidobacterium dominant one often seen in breastfed babies, might actually be optimal for certain aspects of early brain development. The complexity might need to increase later.
Okay, that makes sense. It's about the specific functions and timing, not just raw numbers. And it seems this early microbial environment can even have an impact on the very wiring of the brain.
Yes, studies using advanced brain imaging techniques like fMRI have shown associations between the diversity of an infant's gut microbiome and the functional connectivity, how different brain regions communicate with each other, of specific areas known to be involved in emotional processing and regulation. We're talking about regions like the amygdala, thalamus, anterior cingulate cortex, and anterior insula.
So the gut bugs could influence how those emotional circuits develop.
The evidence suggests that early microbial experiences may indeed play a role in shaping the development and wiring of these crucial neural circuits. It's an active area of investigation.
Now, another significant factor that can disrupt this delicate early microbial landscape is the use of antibiotics. Dr. Martin Blaser has done a considerable amount of important work on this, particularly with his missing microbes hypothesis.
Dr. Blaser's work is indeed very important in this context. His missing microbes hypothesis posits that the widespread overuse of antibiotics, particularly during early life, can cause long-lasting disturbances in the gut microbiome, a state we refer to as dysbiosis, which is essentially an imbalance in the microbial community.
And he argues it's not just a temporary blip.
Exactly. He argues that it isn't just a transient shift, it can fundamentally alter the trajectory of microbiome maturation, potentially leading to the permanent loss of beneficial bacteria that play crucial roles in our health, the missing microbes.
And this early antibiotic-induced dysbiosis has been linked epidemiologically to a surprisingly wide range of health issues later in life.
Unfortunately, yes. We see links to an increased risk of developing allergic and immune-mediated diseases such as asthma, food allergies, and allergic rhinitis. It's also been associated with metabolic disorders like obesity and type 1 diabetes, potentially type 2 as well.
And even neurodevelopmental issues.
There are some emerging links to certain neurodevelopmental conditions like ADHD and intellectual disability, although the evidence for some of these, like anxiety or depression, is still evolving and can be somewhat inconsistent across studies. It's also important to note that the frequency matters. The number of times a child receives antibiotics in those first 2 years seems to matter, with multiple exposures showing stronger associations with these later health outcomes.
And how might these early disruptions to the microbiome lead to these seemingly unrelated problems down the line? What are the mechanisms?
It's likely quite multifaceted. Antibiotics can, of course, reduce the overall diversity of the gut microbiome. They can wipe out beneficial bacterial species that produce vital metabolites like short-chain fatty acids, which nourish gut cells and have anti-inflammatory effects, or precursors to neurotransmitters like serotonin and GABA.
So impacting brain chemistry indirectly.
Potentially, yes. Plus, this disruption can create an ecological niche that can be readily colonized by more opportunistic or pro-inflammatory microbes. This can lead to altered production of neuroactive compounds within the gut itself, increased permeability of the gut lining, the so-called leaky gut, which allows bacterial components like LPS to enter the bloodstream, potentially triggering systemic and even neuroinflammation.
And affecting the vagus nerve signaling, too.
Yes, potentially causing aberrant signaling along the vagus nerve, that major communication highway between the gut and the brain.
So disruptions in that early microbial ecosystem can really have these long-term ripple effects, potentially increasing an individual's vulnerability to mood disorders, metabolic diseases, immune issues, pretty much everything later in life.
Exactly. The microbial and immune programming that occurs during those critical first 1,000 days
Mm.
has these disproportionately large and enduring effects. And given the bidirectional nature of the microbiota gut-brain axis, the gut affects the brain, but the brain, through stress responses, also affects the gut early life adversity, including antibiotic use, can unfortunately set up these self-perpetuating cycles of dysregulation.
It really puts the spotlight back on that early diversity, but maybe more specifically, the identity and function of the microbes that colonize early on, and the timing. It's not just about having lots of different types later, but getting the right foundation.
That seems to be increasingly the view. While overall diversity is often cited, particularly later in life, the identity and function of the colonizing microbes and the timing of their establishment during these early dynamic phases are likely more critical for healthy MGB axis development than sheer richness alone.
Which, again, underscores the importance of things like promoting breastfeeding, using antibiotics judiciously, and maybe even exploring targeted probiotics or prebiotics during these sensitive developmental windows to try and mitigate some of these long-term risks.
Absolutely. That's where a lot of research interest lies now.
Okay, let's shift our focus slightly and consider why we might be witnessing these concerning increases in the rates of allergic, autoimmune, and chronic inflammatory diseases in industrialized nations. The old friends and biodiversity hypotheses offer some really compelling evolutionary explanations for this.
Yes, these hypotheses link the rise of these conditions to a kind of evolutionary mismatch between our modern microbially depleted environments and the conditions under which our immune systems evolved.
Let's start with Dr. Graham Rook's old friends hypothesis. What's the core idea there?
The core idea is that our human immune systems co-evolved over millennia in close contact with a diverse array of microorganisms. These weren't necessarily pathogens causing overt disease all the time, but rather microbes from the environment, our mothers, our families, maybe even low-level exposures to organisms that could cause disease, but often just stimulated the immune system. These are the old friends.
And our immune systems came to rely on them.
Exactly. Dr. Rook talks about an evolved dependence. Exposure to these old friends became essential for the proper development, calibration, and regulation of our immune systems, particularly the branch responsible for regulating immune responses and preventing overreactions, the regulatory arm, involving cells like regulatory T cells, or TREGs.
And TREGs are important for preventing allergies and autoimmunity.
Precisely. They help prevent the immune system from overreacting to harmless things like allergens in the environment, our own body tissues, preventing autoimmunity, and even the commensal bacteria living peacefully in our gut.
But modern life, with its focus on sanitation, hygiene, changes in diet, antibiotic use, C-sections, even how we build our houses, all this has significantly reduced our exposure to these old friends.
That's the crux of the hypothesis. This diminished microbial input during early development leads to an immune system that doesn't get properly trained, fails to develop robust tolerance mechanisms, and as a result becomes prone to dysregulation. This manifests as the rise of allergies, autoimmune diseases like type 1 diabetes or multiple sclerosis, inflammatory bowel disease, IBD, and potentially even the kind of systemic low-grade inflammation linked to metabolic, cardiovascular, and maybe even psychiatric disorders.
So it's not just about missing any microbes, but missing these specific old friend inputs that our immune systems evolved to expect and depend on.
Exactly, an active dependence for proper function.
And the biodiversity hypothesis is related, but maybe broader.
Yes, it's closely related and complements the old friends idea. It emphasizes the importance of overall microbial diversity that we encounter from natural environments. It suggests that our reduced contact with a wide spectrum of environmental microbes, driven by both macroscopic biodiversity loss in our surroundings and our increasingly indoor lifestyles, diminishes the necessary microbial input that's crucial for healthy immune development and the establishment of tolerance.
So less biodiversity around us means less diversity inside us, which messes with immune training.
That's the core concept. The declining biodiversity in the world around us translates to a less diverse human microbiome, which is critical for educating our immune system.
And the evidence from farm studies really seems to strongly support this idea, doesn't it? The lower rates of asthma and allergies in kids raised on traditional farms.
Absolutely. Those studies are quite compelling. The so-called farm effect is thought to be mediated by the increased exposure to a diverse range of microbial components found in those settings, things like endotoxins, LPS from bacteria, fungal components, various pathogen-associated molecular patterns, PMPs, from livestock, soil, and just a generally microbially rich environment.
And these exposures stimulate the immune system in a good way.
Yes, they stimulate the innate immune system and promote the development of those regulatory pathways that dampen down allergic responses and foster immune tolerance.
And Dr. Brett Finley's research has specifically honed in on how these early life microbial exposures influence the development of the immune system and the subsequent risk of asthma, kind of bridging these hypotheses.
His work, particularly the findings from the Canadian Healthy Infant Longitudinal Development Child Study, has been really insightful in this area. They identified a specific microbial signature in infants at around 3 months of age that was associated with a higher risk of developing asthma later in childhood.
3 months, that's really early. What was the signature?
They found that lower levels, or the complete absence of 4 particular bacterial genera, Faecalibacterium, Lactospora, Veillonella, and Rothia, often referred to collectively by the acronym FLVR, were linked to an increased risk of asthma by ages 3 to 5.
FLVR. And it's interesting that these FLVR differences often seem to disappear by the time the children reached 1 year of age, right?
Exactly. That really points to a critical, very early window of influence, likely during immune system programming. These FLVR microbes are thought to play an important role in shaping the developing immune system in those very early months.
And how might they do that?
Their relative absence was associated with altered profiles of metabolites in the infant's stool, including reduced levels of acetate, a short-chain fatty acid, which is believed to influence immune cell development and potentially inflammation in the lungs. Furthermore, analyses have provided evidence supporting a causal role for these FLVR microbes in protecting against asthma-like symptoms in mouse models.
And Dr. Finlay and Dr. Marie-Claire Arietta even wrote a book called Let Them Eat Dirt, which really helped popularize this idea for a wider audience.
Yes, that book did a great job of communicating the science and popularizing the idea that early exposure to a diverse range of microbes is actually beneficial for health, and that our modern obsession with hypercleanliness might inadvertently be doing us harm by depriving us of these essential exposures.
It really challenges the long-held notion that all germs are bad.
Absolutely. It advocates for a more nuanced understanding of the essential microbial exposures that we need, especially in early life. And it highlights how the modern urban environment, with its reduced green spaces and altered microbial landscapes, significantly contributes to this depletion of old friends and overall biodiversity in our immediate surroundings, driving this epidemic of inflammatory diseases.
So the collective evidence from these different lines of research really seems to point towards this critical evolutionary mismatch. Our immune systems evolved expecting a rich microbial world, but now they're developing in relatively impoverished environments.
And this mismatch appears to be a significant factor in the rising rates of chronic inflammatory conditions we're observing.
It's leading to a point where public health officials and even urban planners are starting to seriously consider ways to safely reintroduce or mimic some of these old friend exposures and to enhance biodiversity within our living environments, thinking about green spaces, building design, even balancing hygiene practices.
Exactly. It's a shift in thinking.
This leads us to another somewhat concerning concept that builds on this, the idea of intergenerational microbiome collapse. It's the notion that the loss of microbial diversity we're seeing in industrialized societies might actually be getting compounded across generations.
That's the worrying trend that some researchers, like the Sonnenbergs, are highlighting, that we may not only be experiencing a depletion of microbial diversity in our own microbiomes, but that we could be passing on an even less diverse and less resilient microbial community to our children, and they to theirs.
Sort of like microbial extinction within family lines?
In a way, yes. The research coming from Doctors Justin and Erica Sonnenberg's lab at Stanford has provided some really fascinating experimental evidence to support this idea, using animal models.
What did they do?
They used mice that were colonized with human gut microbiota and explored the effects of feeding them a diet that was low in microbiota-accessible carbohydrates, essentially. A diet lagging in diverse dietary fibers, which is quite representative of the typical Western diet. And they observed some really striking results over multiple generations of these mice.
And what did they find happening across the generations?
Their key findings were, first, a progressive loss of diversity. Mice maintained on this low-fiber diet across several generations showed a stepwise and progressive loss of diversity in their gut microbial communities. Specific types of bacteria, particularly within the Bacteroideales order, gradually diminished in abundance and even disappeared entirely in later generations.
And could they get it back just by eating fiber again?
That was the second key, finding the increasing irreversibility of this loss. While short-term changes in microbial diversity due to the low-fiber diet were reversible in a single generation by switching back to a fiber-rich diet, the diversity loss became increasingly permanent with each subsequent generation that remained on the low-fiber diet. By the fourth generation, simply reintroducing a high-fiber diet was often insufficient to restore many of the lost microbial taxa.
Wow. So they were truly gone from that lineage.
Often, yes, unless they intervened with things like fecal microbiota transplantation, FMT, or the specific reintroduction of the missing microbes from an external source.
So what are the underlying mechanisms driving this intergenerational loss? Why aren't the microbes just passed on?
The primary mechanism appears to be inefficient vertical transmission. That is, the transfer of microbes from parent to offspring, particularly for those microbial taxa that are already present in low abundance in the parental gut due to the low-fiber diet.
So if there aren't many of them to begin with, they're less likely to make the jump.
Exactly. If a low-fiber diet causes certain types of microbes to become less numerous in the parents, they are then less likely to be successfully transferred in sufficient numbers to effectively colonize the offspring, creating this kind of generational bottleneck effect.
And this dietary-driven loss is probably being made worse by other modern lifestyle factors too.
Almost certainly. It's likely being compounded by other aspects of modern lifestyle that disrupt microbial communities and their transmission. Things like the increasing rates of cesarean sections, which bypass that initial vaginal fathical transfer, the widespread use of antibiotics, which can decimate microbial communities, and the fact that infant formula, while a lifesaver in many cases, doesn't fully replicate the microbial richness and complexity of breast milk, particularly those vital HMOs and the direct transfer of maternal microbes it contains.
And we're seeing evidence of this trend in human populations as well, aren't we? When we compare the gut microbiomes of people in Western societies to those living in more traditional communities.
Yes, the comparisons are quite consistent and striking. Studies generally show that individuals living in industrialized Western nations tend to have lower overall microbial diversity in their guts and are often missing certain ancestral lineages of bacteria, like certain species of Prevotella, for example, that are commonly found in the guts of people in more traditional hunter-gatherer or rural agrarian populations who consume diets much richer in diverse dietary fibers.
And even immigrant studies show this.
Yes, studies of immigrants who move from non-Western countries to Western nations often show a progressive loss of their native gut microbial diversity over time, or across generations, which often coincides with an increased risk of developing Western diseases like obesity, diabetes, and IBD. It strongly suggests that the Western lifestyle actively erodes this ancestral diversity.
So this intergenerational microbiome collapse is this cumulative loss of microbial diversity across generations due to our sustained modern dietary and lifestyle practices, which are seemingly at odds with our evolutionary microbial heritage.
That's the concept, yes.
And these aren't just any microbes we're losing. They often perform critical functions that are essential for our health, right? Like fiber digestion, producing SCFAs, immune education.
Exactly. The lost microbes often have critical functions, and their absence can lead to tangible health consequences down the line.
So what are the implications for the health of future populations if this trend continues?
Well, the implications are potentially quite significant and concerning. Each generation might inherit a slightly more depleted microbiome, which could increase their susceptibility to a wide range of chronic inflammatory diseases, metabolic disorders, autoimmune conditions, allergies, and potentially even mental health conditions.
And reversing it gets harder over time.
That's the fear. Reversing this trend may become increasingly challenging, with dietary changes alone potentially being insufficient if the microbes are completely lost from the population pool. It might necessitate the development of novel therapeutic approaches, such as carefully designed consortia of probiotics sourced from more diverse populations, or even the establishment of microbial Noah's Arks, repositories of ancestral microbes that could potentially be reintroduced.
Like Dr. Dominguez Bello's Microbiota Vault initiative.
Exactly. That's a prime example of this forward-thinking approach aiming to preserve microbial diversity before it's lost entirely.
It sounds like addressing this really requires a long-term and multifaceted strategy. It's not just one thing.
No, it includes promoting diets rich in diverse microbiota-accessible carbohydrates, supporting natural pathways of microbial transmission like vaginal birth and breastfeeding, using antibiotics much more judiciously, and fostering environments both personal and public that support microbial diversity. It really calls for a societal recognition of our microbial heritage and a concerted effort to protect and restore it for the health of future generations.
Okay, so given this profound and lasting impact of the early terrain and these potential long-term challenges, what are some of the practical strategies we can think about for nurturing it? What kind of interventions and community support can make a real difference now?
Well, the good news is that because this period of the first 1,000 days is so plastic, it's also a really high leverage opportunity. Positive interventions during this time could have significant and long-lasting benefits for an individual's health trajectory.
So where do we start? Probiotics and prebiotics come up a lot.
They certainly do, and they hold potential. Probiotics, specific strains like Lactobacillus rhamnosus GG or certain bifidobacterium species, and prebiotics like GOS, FOS, or even synthetic HMO mimics added to formula can potentially modulate the infant gut microbiome. There's some evidence, for instance, suggesting potential benefits in reducing the risk of certain allergies, like cow's milk or egg allergy, when mothers or infants are supplemented during pregnancy and infancy.
But it's not a magic bullet, right?
Definitely not. It's important to keep in mind that the effectiveness of probiotics can be very strain-specific, and it likely depends heavily on factors like dosage, the timing of administration, and the individual's baseline microbial composition. We still need a lot more research to fully understand the optimal ways to use these tools effectively and safely in this specific context.
Makes sense. And then there's the environment itself. We touched on this with the old friends hypothesis, but how can we actively modify our environments, homes, daycares, maybe even hospitals?
Exactly. Our built environment, where we spend most of our time, is a significant source of microbial exposure, and we're learning more about how we can design these spaces to support healthier microbial communities rather than deplete them.
Like bringing nature indoors.
That seems particularly important. Promoting microbial diversity through increased contact with nature. This could involve things like incorporating more indoor plants, installing green walls, ensuring access to outdoor green spaces for children, and perhaps even considering more natural materials in building construction.
I heard about studies on rewilding daycare playgrounds.
Yes, there's some fascinating research on rewilding daycare playgrounds with natural elements like forest, soil, sod, and diverse vegetation. These studies showed positive changes in the children's skin and gut microbiota diversity, as well as shifts in certain markers of immune function towards a less inflammatory profile.
That's incredible, even office green walls.
Indeed. There's evidence that even things like air-circulating green walls in office spaces can diversify the skin microbiota of employees and modulate their immune markers.
And what about the materials our homes and other buildings are made of? Does that matter?
There's growing interest in that too. Using more natural building materials like clay and uncreated wood, which can have inherent properties that support a healthier indoor environment, for example. Clay can help regulate humidity and potentially reduce mold growth, while some woods has natural antimicrobial properties. There's even early exploration into the concept of probiotic architecture, though that's still quite futuristic.
And what about ventilation systems and our cleaning practices? We clean to remove germs, but maybe we're removing too much.
That's the balance we need to strike. Natural ventilation, like opening windows, can introduce a greater diversity of airborne microbes compared to tightly sealed buildings, relying solely on HVAC systems, especially if those systems aren't well maintained. When it comes to cleaning, a more balanced approach is likely beneficial.
So less scorched earth cleaning.
Exactly. Avoiding the overuse of harsh antimicrobial products that can indiscriminately wipe out both beneficial and potentially harmful microbes. Organizations like Mom's Clean Air Force and resources like Tiny Health offer guidance on using more eco-friendly cleaners, natural alternatives like vinegar or soap and water for general cleaning, and targeting disinfectant use to specific high-risk situations rather than a blanket approach. The goal is hygiene without oversterility.
What about hospitals, especially NICUs? That seems like a really tough environment to balance.
It's incredibly challenging. In neonatal intensive care units, you have very vulnerable premature infants, and the need for rigorous infection control is paramount. Studies show these environments often have less diverse microbiomes, sometimes dominated by organisms adapted to cleaning agents, like certain yeasts. There is ongoing investigation into controlled trials of things like vaginal seeding or probiotic supplementation in ICUs, but safety is the absolute priority. And it's worth remembering, maternal factors, mom's own microbiome, continue to influence the neonate's microbiome even before hospital discharge.
It really sounds like while individual actions and choices are important, they're likely to be most effective when they're supported by broader community practices and public health initiatives. It can't just be up to individual parents.
Absolutely. That's critical. Supporting and promoting breastfeeding requires systemic support. Family-friendly policies like paid maternity leave, looking at UNICEF and WHO recommendations for duration, providing clean, private, and accessible lactation spaces in workplaces and public areas, ensuring paid breaks for pumping.
And support networks, too.
Yes, running public awareness campaigns about the long-term benefits and offering accessible professional support from lactation consultants and peer support networks like Lalesh League or community health workers, these will make a huge difference.
And promoting healthy maternal diets.
Vital. Public health education on the importance of nutritious, fiber-rich diet for pregnant and lactating women is key, but so is ensuring access to affordable, healthy foods, addressing food deserts and insecurity.
And antibiotic stewardship needs to be everywhere.
Absolutely critical. Implementing and enforcing strong antibiotic stewardship programs in all healthcare settings, hospitals, clinics, urgent care guided by organizations like the AAP and CDC is essential. This applies especially to prescribing for pregnant women and young children. It means prescribing only when truly indicated by evidence, choosing the narrowest spectrum agent possible, the appropriate dose, and the shortest effective duration.
And educating everyone involved.
Yes, educating both clinicians and parents about the risks of overuse and the importance of microbiome health is a key part of stewardship.
And finally, getting back to nature, enhancing access to green spaces.
Definitely. Urban planning policies that prioritize the creation and maintenance of accessible, biodiverse green spaces, parks, community gardens, natural play areas can play a significant role in increasing beneficial microbial exposures for the whole community. Projects like the BUGS project, focusing on biodiversity in urban green spaces, are great examples of community engagement and raising awareness about this connection.
It really underscores the idea that our modern urban lifestyle can often lead to a kind of microbial deprivation that we need to consciously work to counteract. It's not about advocating for unhygienic practices, but rather about finding safe and thoughtful ways for a reacquaintance with the old friends and diverse microbial communities we've co-evolved with.
Exactly. Efforts like rewilding daycare environments, promoting responsible pet ownership, exploring probiotic architecture. These are all attempts to consciously and safely rewild our microbial environments in ways that support health.
And bringing it all together, we can see a multi-pronged approach emerging in Table 2 of our notes, covering birth practices, feeding, targeted supplements like probiotics, prebiotics, careful antibiotic use, supporting maternal well-being, thoughtful environmental design, and supportive community and urban planning. It's a whole system.
It truly requires thinking across all these levels.
So as we bring this deep dive to a close, it's just so clear that those first 1,000 days represent this extraordinary period of plasticity, really laying the very foundation for lifelong health in ways that we're only just beginning to fully appreciate.
Absolutely. The lasting impact of early microbial and epigenetic programming is, I think, undeniable at this point. From that very first breath and those initial microbial encounters, through the crucial nourishment and signaling of breast milk, and even the influences of maternal well-being in our surrounding environment, these early experiences intricately shape our biology and set the stage for our health and resilience, or lack thereof, in the years to come.
And it feels like we're at this really interesting, maybe slightly awkward crossroads in history. The very advancements in hygiene and medicine that have saved countless lives might also be inadvertently contributing to this modern challenge of microbial deprivation and maybe epigenetic disruption, potentially playing a significant role in the increasing prevalence of chronic diseases like allergies, autoimmunity, maybe even metabolic issues.
It's a complex trade-off we're navigating, and the emerging understanding of intergenerational microbiome collapse raises some truly sobering long-term concerns about the health trajectory of future populations if we don't actively address these trends and change course somewhat.
There is still so much more to uncover, isn't there? We desperately need continued and robust research, more longitudinal studies tracking people from birth. work to identify those keystone microbes that play such crucial roles at different stages, a better grasp on the mechanisms of intergenerational transmission.
And, importantly, rigorous evaluation of public health strategies designed to promote beneficial microbial exposures and support epigenetic health in safe and effective ways.
It really calls for a significant paradigm shift, doesn't it? Moving away from just reacting to disease and towards these more proactive, terrain-focused public health strategies. Strategies that prioritize the holistic well-being of mothers and infants, encourage natural and beneficial microbial exposures where possible, promote the judicious use of medical interventions like antibiotics and C-sections, and foster healthier food and living environments for everyone.
That's the holistic vision, yes, focusing on building resilience from the ground up.
So perhaps the final thought for you, the learner, as you process all this, is to really consider this concept of ecosystem management for your own health and maybe the health of future generations. Instead of solely focusing on eradicating pathogens, which is sometimes necessary, of course, maybe we also need to focus on cultivating a thriving, diverse, and resilient internal terrain, both the microbial landscape and the epigenetic landscape.
It's about embracing a more holistic, ecologically informed, and perhaps evolutionarily aware approach to health and disease prevention.
Starting right at the very beginning of life, but recognizing that these principles apply across the entire lifespan. It's a different way of thinking about health.